think we'll just say welcome to Parallell Session B, Innovation Projects, which is in the second seminar in the series of the research in the wake of the pandemic. We encourage you all to use your camera on, but please mute your microphone during the presentations. My name is Unn Sørum and I work with the Trond Mohn Foundation, and I will be the moderator of this session. Welcome to everybody. In this seminar, we want to help to build an academic COVID-19 community, and we thus encourage new collaboration and dialogue between all the projects and the general public. Until now, in the plenary part of this seminar, you have been sitting quietly and listening. But in the parallel session, we encourage you all to be active and please use your camera on. Ask questions, give the questions in the chat, and we will help to forward this to the presenters later on. And please be active to take part in the discussion. In this session, we have asked or invited 2 companies to present their research. You have already listened to them in the 3-minute teasers, which was very interesting in the plenary session. But in this parallel session, they have been asked to give a more in-depth presentation of 10 minutes approximately each. And there will be time for questions afterwards. And I have to inform you that this seminar is recorded and it will be made available at the website for the seminar afterwards. If you do have questions, please present them in the chat and we will forward them afterwards. So then I would like to welcome the first speaker in this session, Per Olav Skjervold, co-founder and chairman of the board of Norimmun, and you have already met him previously, so please, the floor is yours.
Thank you. I'll see if I can share my presentation. Can you confirm that you see my presentation now?
Yes.
Perfect. Okay, thank you. Well, thank you for the opportunity to go a bit in depth describing this project, which is initiated and performed by my colleague scientists in Norimmun and in collaboration with Professor Magnar Bjørås at NTNU and Oslo University Hospital. You see, in addition to this private funding from our company, which was kind of a philanthropic initiative when the pandemic hit us, we have also had support from Forskningsrådet and Innovation Norway to conduct this work. Why can't I? My presentation is frozen. This is not how it should be. Well, okay, sorry, now it works. You see the second page now?
Yes.
Yes, okay. Well, very shortly, so Norimmun, as I mentioned, is a company. It's a private R&D startup company where we have developed and licensed technologies and products to fight viruses, bacteria, and parasites in aquaculture and farmed animals. It has an industrial ownership, mainly from Felleskjøpet, which is a farmer organization. Magnar Bjørås, we met when we took this initiative, and it's been a very fruitful collaboration to us. Magnar Bjørås has a long career, but mainly for this importance for this project, Bjørås Research Group has established protocols and technology for virus neutralization in assays in 2- and 3-dimensional cellular systems. So this has been an extremely good help and active participation. But you see, the upper line here is what we've actually done. What we have done is we have immunized hens, and we have purified IgY from the egg yolk. Then we have used this polyclonal IgY, tested this in vitro by ELISA and in cell-based neutralization tests. And we have just recently performed our first animal trial with hamsters in the Netherlands. So what we aim to continue is, of course, to use this to scale up large-scale immunization where we can use thousands of hens, have an industrial purification, and then pharmaceutical drug formulation. We aim to develop a product that can be administered in the respiratory tract as a prophylaxis or a therapy, and ideally as a combination therapy with already approved drugs, which is, as you might know, one of the focuses on the research groups of Magnus to see how combinations of existing medicines can be effective. As you all know, the vaccine is about active immunization on an immune response in a human to the content of a vaccine. Passive immunization, however, is the transfer of already produced antibodies into an individual for the prevention or treatment of a disease. This is then an immediate but short-lived treatment. And as we discussed, there's two main types of antibodies. It's the monoclonals, which is then one, as I said, a kind of a bullet. The Eli Lilly and Regeneron has already developed this, although they are at the moment hitting some problems because of mutations. It's very expensive production, but it works well when it hits. The polyclonal then is either drawn from human plasma, it's produced in horse serum, or as our approach, it's produced in chickens, and then it's called IgY, which is a different kind of antibody. And I think the most important characteristics in this context is that IgY antibody production is very cost effective. It has a short production time, it's easily scalable, and it's animal friendly, as we don't extract blood from the hens, we use the egg yolk. And in the table, you see the main point of using this table is to show that compared to IgG, it is further from humans, so it's less risk that it can affect unintended immune responses in humans. This is an example of what we do. This is an example from a trial of where we produce passive immunization against the gut disease in poultry. And here from an example where we give this to 10,000 broilers in each group, and in a naturally occurring malaria outbreak, this passive immunization actually works better than a dedicated vaccine. So based on this, we know that the principle works for animals. And here, there's also numerous studies that have shown that IgY protects against the range of viruses in humans and animal models. To the left, it's an example of IgY targeting the Ebola virus tested in mice model, where the upper blue line shows that it gave 100% protection, whilst the control group all died within 10 days after the virus. Whilst on the right, it is an example from a human trial here, protecting against diarrhea in children, and also showing a good effect, but again, highlighting that there's no adverse events due to unintended immune responses. So these are from our own research. This is ELISA from, on the left, it's 2. We see that IgY shows high affinity towards their respective antigens. On the first, it's the spike 1 antigen, and the lower is the remind binding domain of the SARS-CoV-2 virus. To the right, you see a curve where we also have a lysate from the actual virus, and we also see a good binding towards this. This is probably the most interesting curve, because first, already last summer, we did the first trials, and we showed that the IgY antibodies had shown strong virus neutralization of the virus in vitro. But then we continued, and we have now developed combinations of different anti-SARS-CoV-2 IgY antibodies, which further increase neutralization potency. So you see on the curve to the left, we can have lower concentrations on the upper curve, which is a combination to reach 50% protection. Compared to other antibodies and what is published on IgY in this context, we see that our concentrations is very potent. So we are in line, or actually quite better than what's published by others. But also compared to the monoclonal antibodies, of course, we need a larger volume of antibodies, but that's due to the fact that it's polyclonal. So as I mentioned in the previous talk, it's more like a shotgun, that they are weaker, but there's a lot more, and they are maybe better suited for a moving target. Summarizing the differences between the monoclonal and the polyclonal, the reason why we use this is, of course, that the monoclonal is already used and approved in human trials. As we mentioned, President Trump got this. He got the monoclonal antibodies and already approved treatments, which is maybe the main idea of our approach also. But what we can do is we have a low-tech, low production cost, more fast production, and potentially less vulnerable to mutants. And that is what we have not done, but that we are into looking now, that we will start trials in vitro and also in different in vitro models and also animal studies to see how if this is more robust, also our treatment is more robust to mutations of the virus. So this project, which is funded by the Research Council, has only a 1-year duration. I've worked with science-based innovation all my life, but never with such a rush. So what we started before the project was funded, we established a new animal research facility. We selected some SARS-CoV antigens and developed an immunization protocol. So early this summer, we were able to purify antibodies. And during the summer, we had the first proof of principle studies in vitro wild-type neutralization of the SARS-CoV virus in Vero cells done by Magna Bjørås. And then we have just recently performed our first animal study, and we will continue to do more animal studies. We haven't got all the results yet, but we clearly see indication that this works as intended. We are looking into, as mentioned, mutant virus neutralization, and validate formal results in other cell types and organoid systems, also having more complex structure with different cell types. We will work further on combination therapy with approved drugs, but we will also continue the work to further increase the potency and product optimization with other IgY types. And last, we will also work on a more cost-effective antigen production for this upscaling technology. So my last slide is summarizing what are our ambitions beyond this R&D stage or this project stage. Of course, it's to develop production platform for industrial scale IgY production in Norway, exploiting the unique potentials for rapid production of high volume, low cost IgY. The second is that our intention is drug development through licensing or partnering with biopharma industry. And the third is contribute to fight COVID-19 in addition to future pandemics as part of a Norwegian strategy for pandemic preparedness and risk management. So that was my last slide. Thank you for listening, and thank you to my colleagues, scientists, for the work that they've been conducting the last year. Thank you.
Thank you, Per Olav Skjervold, for a very nice presentation. Are there any questions? Please add your camera on if you feel like it. Any questions from the audience? Yes, Tina Torbjørnsen.
Have you said that you want to license this technology to pharma companies? Are you in contact with some of them, or have you some reactions from them?
So first of all, I can answer, but if it's more technical questions, I think several of the scientists are also included in the Teams meeting, so they can help me. But no, we have not done that at this moment. As I said, we have similar experience from the veterinarian side, and to this stage, I think we are getting very close to that point now, because we had to document well enough that this actually works, and also we need to do the patent protection, because that's how life works. If someone wants to invest huge sums to get this approved to the market, it has a cost, and then you need some kind of protection. But we have patent applications, so we hope that's also in line. So hopefully during the coming months we'll establish that contact. But I'm very honest to say that we have a very strong network in the aquaculture and the veterinarian industry globally, but we do not have in the biopharma. So we are very happy if for any collaboration to get the right contacts.
Could you tell me, I have one question. I'm not really an expert, but you have mainly focused on the veterinary medicine, of course, until now, but are there any other examples from throughout the world with experience with the use of avian IG?
As I said, I had to go rapidly through all the presentation, but you saw it. There's a lot of publications shown both in animals but also in humans, and there's products used. For instance, in Asia, there's a lot of products like the one you saw the example for gut disease, because this is administered either as an oral treatment or as injections, or like tested in Sweden a few years ago for in the respiratory tract for people with severe lung diseases. Mm-hmm. Other examples, but there's a lot of patents. There's a lot of scientific reports, and there's some products. But as mentioned earlier, I think there's both the culture and the upscaling question which has limited the use of this.
Mm. Any questions? There is a raised hand, Lina Rodal.
Yes, I didn't hear that you mentioned it. Maybe you did, but if you inject these IgY, how long would that effect last? Seen, it's shorter. So how?
Well, maybe two comments from me. First, our intention is not to inject. Also, the monoclonals, also what Trump got was an injection, and this is both very expensive, the monoclonals, but you have to be on intensive care at the hospital, so it's a very other areas to give this in the respiratory tract as a nasal or a mist, and then it's a lot easier to do it. But what the monoclonals, the approval or the temporary approval for the monoclonals from Lilly is then to give this to patients that have tested positive, but is not severe ill. So it's in the first, let's say, 10 days. And what they document is that it's very reduced frequency of ill, also that people have to be hospitalized with severe injuries. So it's not to prevent, which is also an option, but it's to reduce the risk of severe injury. So it's days, the duration.
Okay, so when you get it in the respiratory tract, it lasts for days.
Well, my colleague scientist again can help me. We don't have the trial results from this case, but the principle, of course, is that you don't. This is well, we see from other when we do this on animals that it can have a longer duration, but in principle, it's not renewed by the patient, so it has to be done given a repeated like a medication that you have to continue for a while.
My name is Gorm Sansom. I'm a veterinarian working for this project. I could give a short comment here. Based on the phase 3 studies that has been performed in humans, where it's been oral application of IGY, is usually being repeated on a daily basis, which means that they reckon that the pharmacokinetics might be half-life of less or of 24 hours, not much more at least. So around 24 hours half-life is what they expect in a local application. There is, as far as I know, no licensed IGY preparations for injections, but that might be wrong. But I don't know of any that's been recognized for injection applications of IGY for the moment. So there is, as far as I know, no pharmacokinetic data for humans for that. But of course, there's been performed in a lot of animals, so there you can find pharmacokinetic data. Yeah.
Okay, I think we should move on, and thank you very much, Per Olav. Maybe there will be more questions later on. This room will be available until 12 or so, if there is more questions coming up. But I'll thank Per Olav for a very nice presentation and nice questions, and I will welcome the next speaker of this session, medical doctor and head of clinical development, Katrin Lund Hadley at H Labs AS. Please, the floor is yours.
Thank you so much, Un. Can you see my screen?
Yes,
yeah.
I can.
Okay, perfect. Okay, I'll talk about our project Predict COVID, or PredCov. H Labs is an Oslo-based diagnostics company. What we do is we apply machine learning to big epigenetic data sets from biobanks to develop diagnostics that we in turn turn into actionable blood tests for clinicians, researchers, and individuals. Our strategy is to measure the process leading to the disease instead of the disease itself, as this enables us to detect disease earlier. Our main focus is on age-related diseases. In this COVID project, we have teamed up with the Norwegian Institute of Public Health, Oslo University Hospital, and Bærum Hospital to develop diagnostics for COVID-19. We are a multidisciplinary team of molecular biologists, a veterinary, biostatisticians, and medical clinicians. And we have received funding from the Research Council, for which we are very grateful. One of the challenges with COVID-19 is the diversity of complications and how different people respond to the disease. While some people experience mild symptoms, others develop severe complications rapidly and need intensive care. And this is the reason we started this project, to develop diagnostic tools for doctors to guide treatment. One of the backgrounds, or one of the reasons we looked into this also, is that we know from previous research that specific changes in gene regulation can be used to predict severity of viral disease and also who will respond to vaccines. And we believe that these signals can be used to predict COVID disease severity earlier, enabling us to prevent deterioration instead of treating complications when it's too late. So our main aim in this project is to predict COVID-19 severity to enable earlier and targeted treatment for those at risk. For example, it would enable clinicians to give steroids and also predicting and diagnosing long COVID. Our method is to analyze the switches that determine which genes are turned on and off in white blood cells, and this is called DNA methylation. And these changes often precede of other conventional biomarkers, making it possible to detect signals much earlier, while conventional biomarkers often measure the result of the disease. This can be measured earlier. And as mentioned earlier today, these switches may change in response to environmental factors as well, such as viruses. What we do is we analyze 850,000 epigenetic markers in every blood sample, and this covers 99% of all of the human genes. Our general approach in this project is first to develop an algorithm where we apply advanced statistics and machine learning on large epigenetic data sets of more than 500 COVID patients and controls. And then we use the CpG sites we have found to develop and optimize a lab method. And then afterwards, we have a clinical validation study that will be run later this year at Ullevål and Bærum Hospital, where we test how the test performs. Our project includes 3 cohorts. The first one is the post-COVID and long COVID with 200 patients and controls. The next one is the hospital cohort, and we also have an outpatient cohort. So the results I present today is in the post-COVID cohort. So what we did in this first analysis is that we included 96 cases from the Corona study and 96 controls. Half of the cases had a mild disease and half had moderate or severe disease, defined either as hospitalization or self-reported severe dyspnea and high fever above 39 degrees. The control group consisted of healthy controls, but also a group that had reported symptoms of upper respiratory tract infections, but they had tested negative for COVID. All controls had negative antibodies for COVID to ensure they were not false positives. All participants donated a blood sample about 6 to 12 weeks after their disease. As for demography, there was a similar gender distribution, and the age distribution covered most age groups, not children, though. So first, we performed an epigenome-wide association study where we compared all the post-COVID cases against all the controls. We then found 2 significantly hypomethylated CpG sites. One was on the gene IFI44L on chromosome 1, which is linked to Sjögren's syndrome, SLE, and Kawasaki's disease, but also in general to naive CD4 cells, which could also be an explanatory. The other significant CpG site was on an unknown gene on chromosome 18. We haven't found any info on this CpG or gene on PubMed, Gene Omnibus, or Google. Also, the changes we saw, they correlate with disease severity. The next analysis we did was to compare mild versus moderate and severe COVID-19, where we found significant changes in the CpG site on SEC14L1 on chromosome 17, which is linked to asthma and allergy atopy. This is the one hypomethylated CpG site. So as for conclusions, the differentially methylated CpG sites between all COVID-19 patients and controls suggest that the disease induces these changes in methylation. These findings are preliminary and must be interpreted with caution. We are writing our first paper right now, and it's not been, so we haven't had it peer reviewed yet. It remains to be seen whether these changes are transient or if they may trigger autoimmune diseases in susceptible individuals later on. We have not studied children. However, it might explain why some children develop Kawasaki's disease after a COVID-19 infection. As for the difference between mild and severe disease, the CpG site linked to asthma may well be due to innate susceptibility. In other words, that these patients had a light or undetected asthma prior to the infection that may in part explain why these patients had a severe disease. These are hypotheses. Our next steps in this project will be to analyze long COVID to see if there are changes in certain genes that correlate and maybe may explain the tiredness and memory loss experienced by 10 to 30% of patients after COVID-19. And the interesting thing about long COVID is that the fatigue and memory problems occur regardless of disease severity and age. Also, we will focus on study blood samples taken early on in the disease course to find predictive biomarkers for COVID-19 outcomes. And that was the results I had. If you are interested in collaboration, please reach out. And I want to thank our multidisciplinary PredCop team, which have done a fantastic job. And thank you all for the attention. Thank you.
Thank you, Catherine, for a very nice presentation. Are there any questions from the audience? Lina Rodal, please. You have to turn the mic on.
Sorry. It was a really interesting presentation. So thank you very much. I'm wondering if I'm not so familiar with methylation because I've never actually read so much about it. But is it so that this can be reversed, or is it an effect on the genes that will be there forever, or is that anything known about it? That how that will be?
We don't know yet for COVID. We know that some viral infections cause transient changes, while others can induce changes that are lasting. And that is why some viruses can induce cancer or other diseases. So some are transient, others are not, and we don't know for COVID-19 yet.
But is it, if you see some of the patients that got this AstraZeneca vaccine, they got really severe by side effects. And is that something that you also see in correlation with this methylation? That or is that?
I believe that was auto antibodies, right? That they think was the reason. We haven't studied AstraZeneca cases, not yet. So I don't know.
No, I just mean if it's some of the same effects that happens in the body.
It may be.
Yeah. Yeah.
But I'm sorry, I don't know enough about it at this point. But it's a good question.
More questions? Tine Torbjørnsen, please. You have to turn on the microphone, please.
Maybe I didn't get all your information, but how can you translate the methylation pattern into treatment?
That is a good question. As that it is a field where people are trying to change the DNA methylation to treat diseases, and that would be great because there is a link. For example, diabetes, type 1 diabetes. There are research groups working on medications that can change the switches and then maybe cure type 1 diabetes by using an epigenetic drug. But I haven't seen anyone who have developed this technique to a stage where it actually works. Another thing is where you have DNA methylation at the top, and then you have transcriptomics and proteomics. So it is possible to look at proteomics downstream and use, downstream, and then use those proteomes to as a basis for drugs. That is possible. And we have looked into that, to doing transcriptomics as well, but in the first phase we are focusing on DNA methylation. But for drug targets, that's a very good question and a good idea.
Any more questions? No. Any final comments then from the team, from the partner listeners? Okay, then Per Olav, your microphone is not on. If you're talking.
Yes, yes, should be. Yeah. No, I don't have. I'm fine. So.
Okay.
Thank you for the opportunity to present. So.
Yes, thank you for the opportunity to present, and it was interesting to hear all the talks today and to learn about other aspects with this pandemic, not only the one we're working on ourselves.
So then,
and we will
follow. Please.
And we are interested to follow your project, so we will look forward to new information from your companies.
Thank you.
Thank you.
Okay, then I will thank everybody for taking part in this parallel session, and there will be the third seminar in this series will be later on. I don't recall the date. Amanda, you? No.
No date yet, September
No.
so far. So.
But information will come, and we will come back to you with the dates and program in time. Okay. Thanks a lot.
Okay.
Thank you.
Thank you. Bye-bye.
Bye.